He has 2 postdocs focused on protein-based molecule discovery using interatomic and chemoproteomic. He is expert in confocal microscopy, flow cytometry, and mass spectrometry, specialist at Butantan Institute, leading the chemoproteomic platform for novel molecular target discovery. Thank you, Marcos, for your acceptance to give this lecture. You can start now.
Thank you. Can you see my presentation?
Yes.
So thank you for the invitation, and thank you. It's a pleasure to be here talking about this piece of work that we've been developing in Butantan Institute. So I'm going to talk a little bit about this molecular target discovery that we've been doing at Butantan Institute. So that's almost
recording in progress.
That's almost a technology dogma that venoms are complex mixtures comprising small organic molecules, amines, peptides, carbohydrates, lipids, proteins, enzymes, etc., with huge therapeutic potential. So you know that while it can kill when you get bitten or in the accident, it has a lot of medical potential within the venoms. So it's a classical rationale that to find and develop drugs based on animal venoms. And mainly how we do that is by taking the venoms, we purify molecules from the venoms, and then we improve this molecule by chemical or some kind of computer things with the IA, for instance. Then we have the drug. And we'd like to do the same thing here in Butantan in the past. So what do we think about it? We know that accidents with Lonomia obliqua triggers an hemorrhagic syndrome. So as you can see here, Lonomia is this kind of caterpillar here, and when people touch it, it causes this coagulopathy that can even lead to that. The Lonomia obliqua is the only worm that we know that can lead people to death. So we'd like to understand this kind of envenomation by Lonomia obliqua, and then through venom purification, transcriptome analysis, proteomics, and bioinformatics, we found 2 procoagulant proteins. The first one was this one with these 4 domains, G1, G2, G3, and G4. And when we compare the structure with other proteins, we know that it's a hemoline-like protein. But it really activates factor X. So it was one of the molecules responsible for this procoagulant activity. But other studies suggested that this protein has this neuroprotective activity as well, and also it has an anti-apoptotic activity. So it's not only an factor X activator, but also it has this protective activity as well. And this protein was called LOSAC. And the other really important protein studied in the Lonomia obliqua venoms was this one, which is a lipocalin, and it was thought to have this prothrombin activator, and it also has some cytoprotective activity as well, and it was called LOPAP. So we'd like to improve this molecule. We know that lipocalins and hemolines has this protection. It is involved in development, regeneration, tissue repair, cell growth, and differentiation. But we didn't want these coagulant effects. And how could we remove these coagulant effects? By trying to find the peptides that was responsible for this other activity. So that's what we have been done. But the thing is, when we thought about having these cytoprotective peptides, we didn't want only to have the peptides. We would like to use it as a tool. So the classical way to work with the venoms and peptides, which was like having the compounds, do the characterizations, do the activity, then do the rationale and design of the peptides, find those peptides. But then when we have these peptides, why not trying to find these candidates, use those peptides, the best candidates for target discovery? And because of those proteins has also this neuroprotective, cytoprotective, anti-apoptotic activity, we'd like to understand and use them if they could be used against osteoarthritis, which is a multi-cellular disease. So that's what we've done. So from all the domains of those proteins, so for each domain, for the G1, G2, the 3, and the 4, or from the beta helix, the alpha helix, or the beta sheets from the other proteins, we develop a lot of peptides, and then we started screening all of those peptides to test whether is the one responsible for this anti-inflammatory activity in the osteoarthritis. As I told you, we'd like to understand the participation of those peptides against osteoarthritis. So first, for the screening, you had to choose cell models that participations in articular inflammation. And when you look for a knee, for instance, you have some sensorial neurons that they receive, process, and conduct sensorial signals to central nervous system. We also have synovial that control the synovial liquid composition and extracellular matrix. We have the chondrocytes that produces and maintain the cartilage matrix thick and organized. And also we have the macrophages that maintain tissue hemostasis with innate and adaptive immunological system association through interaction with secreted cellular mediators. So we have tested in all those cell models, and for the readouts, for the screening, we tested the cytokine release, we also tested the neuropeptide release, and also we investigated the inflammation activation. So that's our workflow. So for the neurons, as I told you, we'd like to have this anti-inflammatory activity. So first you need to provoke the inflammation, or in the case of the neurons, we'd like to provoke the pain. So our group has published this model, which we treated sensorial neurons with glycated collagen. Then we put the peptide, and we analyzed the pronociceptive substance P and the beta endorphin release. For the chondrocytes, then we treated with IL-1 beta, which is a pro-inflammatory cytokine. Then we put the peptides and investigate the release of IL-6, IL-8, TNF-A, which is pro-inflammatory cytokines. And for the THP1, we used LPS, which is a pro-inflammatory molecule from most of the microbials. Then you put the peptide, and then we analyze again IL-6, IL-8, which are pro-inflammatory cytokines, but also IL-10, which is an anti-inflammatory cytokine. Also investigates IL-1 beta and TNF-alpha. And after doing a lot of work, we also did a lot of high-throughput analysis with high-content screening imaging, multiplex for other proteins investigation. We also did some transcriptomic, proteomic, and also ELISA. And why would that? Because we'd like to know only the best candidates for target discovery. We have screened more than 50 peptides and other molecules, and only 4 of them were able to be used as tool for target discovery. So that's the main effect of those peptides. From LOSAC, we have the L6 and D3. From the low PAP protein, we have the P4 and E8. And as you can see in this table, for instance, L6 in THP1 using LPS was able to enhance the expression of IL-10, which is an anti-inflammatory cytokine, and it's really rare to happen with peptides. But in chondrocytes, stimulating that with IL-1 beta, we could observe the downregulation of IL-6. And P4 peptide had a really good results in several models. In chondrocytes, for instance, it reduces IL-6, IL-1A, IL-8, GCSF, TNF-alpha. And for neurons, it reduces substance P and releases the beta endorphin expression. For the inflammasome, it inhibits the inflammasome by NLP3, caspase 1, and AXA expressions, but also downregulates IL-1 beta while raising the expression of IL-10 with downregulation of NF-kappa B translocation. For the G3 peptides, again, in chondrocytes, it downregulated IL-6, IL-1H, GSSF, TNF-alpha, GM-CSF. each downregulated in THP1, which is a monocyte, IL-1 beta, IL-1 6, TNF alpha, and in neurons, each downregulated substance P and enhances the beta endorphin expressions. And the EH peptides in chondrocytes, again, a lot of downregulation with pro-inflammatory cytokines, while downregulating substance P and enhancing B endorphin expressions. So those 4 peptides were really good against osteoarthritis, but the thing is, we didn't want to have those peptides as a drug, as a product. We would like to use them as a tool for target discovery. So how can we do that? And that's how my work started, doing the chemoproteomic for target discovery. For doing this proteomic, mainly we fish the targets within the pool of cells. So what's the rationale? So we have our peptide, our molecule. We treat the cells that we have, the model that we have, we just treated the peptides, and then we have the peptides to interact inside the cells with the protein target. Then we have to cross-link and bind it covalently, the peptides within this target. Then use some bead. We purify those complexes. Then by mass spectrometry, we can finally find the target identified. So in order to do that, we need to do some peptide modifications. So if you have any peptide here, you can have any sequence. You can do that by the N terminal or by the C terminal. We have both syntheses happened, but most important, you have this photoleucine group, which has this diazurine reactive group. This diazurine, upon UV excitation, it forms a free radical that interacts with mainly carbon, which has hydrogen nearby, so it can interact with any protein, nearby protein. We have this PEG-4 spacer just to have a separation for the biochain that we use as a tag. So the biochain here is used for purification tag or for labeling tag that I'm going to show you. So how it works, our project. So first, the protein interacts with the peptide. Then by UV excitation, we make a covalent bond between the peptides and its target. Then we can use streptov gene labeled with alexafluor 4-HH. So now we can see if our workflow is working. So it was. So as you can see on the left, we have the native peptide without any signal for cross-linked proteins, and with the modified peptide, then we can see some proteins that are being labeled with this technique. And also, you can see within the cell by using a confocal microscopy that the peptide is inside the cell and labeled with its target, that we cannot identify them by this technique, but you can see that our technique is working, that the peptide is labeling its target and it's inside the cell. So for purification of these targets, then we used a bead conjugated with streptov gene. It's a girl's bead conjugated with streptov gene. Then after lots and lots of washings, then we have only the peptides and the beads in our tube. Then you do this digestion on bead, so you don't interfere with the quantification of the beads. Then you have the peptides, and you go for the mass spectrometry workflow for the protein identification. But one thing that can be a problem is the misidentification of the targets. So to avoid that, we did this competition assay. When you do a native peptide, we do treatment of the native peptide of the cells. Then you do the treatment of the cells with the modified peptide. Then it enhances the amount of native peptide to act as a competitor of these interactions. So what do we have? When you have the native peptide, we almost cannot identify and quantify the protein. When we have only the modified peptide, we should have the most intense ratio of this target identified. And when you have 3 micromolar of the native peptide, for instance, we want to see a smaller signal. As we enhance the amount of native peptide, we lower the intensity of the target identified and quantified. So that we have done, that's our workflow for this chemoproteomic assay. And that would call a specific target. So through bioinformatics, we do the identification, quantification, PCA, clusterization, then we find the specific targets. As you can see here, it's our real data. We have these for all 4 peptides. We have these also in the 4 out of our models. As you can see, some of those targets are not specific. They are just washed away from our analysis, and use only those in red for the, as we can call, specific target. But there were a lot of specific targets. For in this example, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 targets. We could not test all of those for the target discovery. So what we did, we went through literature search. I did not tell in the beginning, but we have a lot of collaboration with GSK. So they taught us a lot of how to work with the drug discovery, and they asked us if there were any other clues about the target, and if the thing was yes, so we could continue on it, but there are drugs being developed with those targets. If the answer was yes, we need to skip that. Are there patents? The same thing, you need to skip that. And there are genetic correlations of these target with the disease, and if the answer now is yes, then you have to go for the validations. And we did a lot of validations. We did knockout cells, and I cannot tell you about the targets because we has signed an NDA with GSK, but I can tell you the results. So for instance, in neuron cells, when you knock out this target using the EH peptide for the activity, when we have the white type cells, the EH peptide enhancing the beta endorphin release. When we have the cells without this target, the EH could not anymore enhance the release of beta endorphin. The same thing happened with the substance P. So in the wild type and with the empty vector that we use for knockout, and the EH could downregulate the substance P, but when we have the knockout cells, the EH could not downregulate anymore the substance P release. So that's for our conclusions, that the rational design of peptides based on low-papi and low-Zac proteins allowed the discovery of anti-inflammatory and antigen-susceptive peptides without this procoagulation activity, and the chemoproteomic of modified peptides allowed us to identify the targets of those peptides. We proved that at least one of those targets is responsible for the anti-nociceptive activity of these peptides, and also that the new target is not described to have participation in any pan-molecular pathways, and it's already being used for drug development. That's why I cannot tell you which target it is. So I would like to thank you for your patience. I thank you for the organizer for inviting me. We have a lot of people working in this project, so I'd like to thank them all. And also I cannot not thank all the funding agencies, the GSK, Butantan, Fundação, the Centro de Desenvolvimento e Inovação, the LDI, and FAPESP. So thank you.
Thank you, Marcos, for your amazing presentation. Great talk. Some questions? Someone has a question? Sorry, what's your name? Gloria. Marcos, Gloria will make one question. Dr. Gloria.
Thank you. I don't know if you can see me, probably not.
Yeah, I'm struggling here with the Zoom, but I'll try.
Yeah. Thank you for your amazing presentation. That's so nice. You showed a reverse mechanism to find targets for putative drugs. That's so amazing. It was like very, very, very interesting. And then you showed that using competitors, they are very, very specific. But I couldn't catch exactly what kind. The kind of competitors. You said that the native peptide. What do you mean by native peptides? Because you didn't modify, you just digested the proteins, and for me they have the same peptides as the native proteins. I didn't know what you mean by competitive native.
When we use them, we modify the peptides so it can bind to the targets, right? So and the modified peptides, they bind the targets, they have this biochain for purifications and everything. When I say native peptide, it doesn't have any of those modifications. It only is the pure peptide. So my competitor is the peptide without any modifications. The rationale behind it is that the peptide competes for the same binding site. But without the diazerine and also without the biochain, it cannot be used for purification and binding and purifications. So if we compete allosteric with the same binding site, because the molecule is quite the same. I don't know if you understood.
Yeah, I understood. But even using this strategy, you may have some false positives. Do you calculated the amount of false positives you had in your experiments? Because you used a lot of peptides, and then maybe you can calculate I don't know how, but the false positives.
Because we tried only 4 peptides, not that much, because the 50 peptides that we tested was only to test which of those has the best results in reducing anti-inflammatory activity, but only 4 peptides were used for chemoproteomics. But we used the FGR for calculation of the unspecific targets, and because what we think we have the empty column as well, and everything that bound to the empty column, we just wash it. And also the most important thing here is that we need to have this behavior, and after all of these we had to check if these were real or not. So we did a lot of tests that I did not show you, but I have a gel with cross-linked, and by using antibodies against the target and labeling the peptide as well. So we have you can see a superposition of it, and most important, we did the knockout of those cells, and you see that with the knockout, the cells loses the activity. So yeah, we can have these false positives, but we took a lot of care before doing the validations.
Thank you. Congratulations.
Thank you.
Marcos, Professor Maria Helena de Lima will talk to you.
Thank you.
Thank you. Very nice, Marcos. I think I did catch which cross-linker did you use?
Use a diazerine. So what we've done, when you synthesize the peptide, we put a photoleucine on it. So the photoleucine, instead of have leucine, it has a diazerine, which is a UV cross-linker. When we put the peptide with the cells, then we stimulated that UV 375 nanometers, 5 joules per centimeter square. You can have the cross-link to happen inside the cell, wherever it is. So the cross-linker is synthesized within the peptide.
Okay, because I used some in the past, I think as derived from azida, but I don't remember exactly. You bind the peptide with your reagent, and after you bind it to the membrane, for example, you stimulate by the radiation, no? It's the same principle.
Yeah, the thing is that the diazerine is already in the peptide. I do not label the peptide, the synthetic peptide. I just order a synthesized peptide already. So it's a little bit different, but you can do that without no problem. If you use some SJG reagent that for chemistry, just label your peptide with there's some cross linkers there, you can do that, and then you use those diazurin for cross-linking, no problem at all. But the problem is that you have to have this biotin tag or some way to purify your complex. That's why when we rationally designed the proteins, we thought, no, just have to have this modified peptide synthetic. We didn't want to label the peptide, then miss something, so we decided to go for the synthetic peptide modified already.
Okay, but your linker cannot be so big because you can have the
Yes.
hindrance, the problem, so what size are these reactivity do you use?
The cross linker that I showed you, just the size of a leucine, because it's a photoleucine. Okay. So just the same size as an amino acid.
Okay, not so big. Thank you.
Mm-hmm.
Anyone else? Marcus, thank you so much again for your great presentation. And I thank you, the whole audience, people that are here present and people that are seeing our transmission online. Thank you very much. And we finish this section. And I suppose that we finish all the activities for this day. Then we see you tomorrow. Thank you.
Thank you.